From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. This episode was recorded on April 18th, 2024. Hello, Micronauts, and welcome once again to today's Quality Quorum. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. Today, believe it or not, is the 37th episode of Matters Microbial. Those of you who watch and/or listen and spread the microbial word to others, thank you. This is indeed a microbial dream come true for me. I think that most of you know how I feel about fossils. A couple of years ago, my wife and I traveled to upstate New York to Beecher's Quarry, which contains ancient pyritized fossils that preserve delicate body parts of things long dead. Here is the last of the fossil trilobites that I excavated during the visit, expertly prepared by Marcus Martin. Amazingly, no relation. As you can see in this photo, visiting the quarry and finding my own fossils was a peak experience for me, and yes, that is a fully genuine smile on my face, as I often hear. I will put links about trilobites, Beecher's Quarry, and an article about Marcus's work studying trilobites in the show notes, and they're very much worth your time if you have those interests. In addition, I wanted to tell viewers and listeners about the wonderful microbial glass art of Jane Hartman. Here is a glass microbial pin I'm wearing during this podcast, as well as a glass sculpture of a bacterium that I cherish. I'll put a link to Jane's wonderful artwork in the show notes. My introductory biology class is drawing to a close. It's pretty emotional for me since it's the last time I'll be teaching that class as I begin phased retirement in the fall for 2 years. Still, I do my best to introduce my students to interesting ideas, to get them thinking and learning and exploring. Metagenomics is a fascinating subject. It's the study of all of the DNA from a given environment. My friend and colleague Joe Handelsman gave me a sample of a metagenomic library made from a soil sample taken from a Puerto Rican cloud forest many years ago. Each bacterial cell in that library contains a phosmid with an insert from some kind of organism in that environmental DNA sample. So I asked my students to test to see if any of the phosmids in that library contain antibiotic resistance genes within their inserts. Since people have never lived in the cloud forest, it's an interesting question. And indeed, you can see that my students have found colonies resistant to the antibiotic kanamycin, the antibiotic spectinomycin, and the antibiotic rifamycin. And they were also able to find clones containing a lactose-degrading gene, which forms a blue colony. Now, over the summer, I'll be getting DNA sequence from these interesting phosmids, and my microbiology students in the fall will find the genes within the inserts by bioinformatics, compare them to other genes, and perhaps identify the organisms from which those genes came. I'm going to put a link up to a very nice introduction to metagenomics in the show notes, and I should also point out emphatically that Joe Handelsman first coined that term. Today's podcast session is particularly interesting to me. Wolbachia is a symbiotic bacterium found in over 80% of arthropods. Ed Yong, the famed science writer, calls Wolbachia the most successful pandemic in history. And Wolbachia does very interesting things inside of the host cells, changing them in many ways. So my symbiotic fascination is intensified. Thus, it's a genuine honor to introduce Professor Sarah Bordenstein of Penn State to this Quality Quorum to tell us how she uses the Wolbachia system to teach young people about molecular biology, bioinformatics, and evolution. Sarah has won awards for the program she administers and champions, Discover the Microbes Within. Welcome to our Quality Quorum for today, Sarah. It's great to see you.
Hello, thank you. It's so wonderful to be here, Mark.
Well, this is something I've really been looking forward to. So I think the first thing, if you don't mind, is give kind of I don't know where the expression thumbnail sketch comes from, but I think you know what I mean. Tell folks about Wolbachia.
So, Wolbachia is something that it was actually discovered 100 years ago, exactly 100 years ago. So, it was discovered in the reproductive tissue of mosquitoes. At the time, it was thought that maybe all arthropods have these rickettsia-like organisms within their tissues. However, we now know that it's found in about half of all arthropods. But when you consider that there's over 1 million named species of insects alone, and some people think there could be 5 to 10 million insects that are waiting to be discovered. So if we have about millions of insects, half of those have Wolbachia, it's incredibly abundant throughout the world. We can find it in every environment where you find arthropods. And by arthropods, I mean the insects and the spiders and the crustaceans. And the mites. So within all of those, you can have Wolbachia. Now, as you said, Wolbachia does some fascinating thing to its hosts. The biology of Wolbachia is pretty cool because it is a reproductive manipulator. So as you can see here, it can induce male killing where the mom will still lay male and female offspring, but all the males are unfortunately killed, allowing the females to survive. It can induce parthenogenesis, where females can pretty much make clones of themselves, and you have all female populations. It can also induce feminization, where mom will lay genetic male and female offspring, but the Wolbachia allows for those males to develop into fully functional phenotypic females. So throughout all of this, Wolbachia is really increasing the amount of females in a population, and it's preferentially selecting for females. And that's because it is passed from mom to offspring. So here we have a bacteria that's inside the cells of the insect's reproductive tissue, and it's only passed from mom to offspring, similar to mitochondria. So when you have that, the male's a dead end for this microbe. So it has evolved these cunning strategies to be able to allow itself to pass on. And then one of the core, the fourth phenotype that we study in our lab is called cytoplasmic incompatibility. And this is not, in this case, it doesn't select for females, but rather it selects for infected individuals because it manipulates the sperm of males such that it can only reproduce successfully with an infected, similarly infected female. If that female doesn't have the same Wolbachia infection, those offspring will die. So over time, if that infected male can only mate with a female that's infected, over time you now have this propagation of Wolbachia throughout the population. Yes.
Sarah, I had a question about the kind of gender bending aspects of Wolbachia infection. If there's a move toward the population being female, do males slip through the cracks of the system, so to speak?
That's a great question, and that's something that people often ask because how do you sustain a population? It should just go extinct if all of the males are gone, correct? So the Wolbachia, it's not always 100% efficiency, and so there can, maybe instead of having a 50-50 ratio, it will kill 80% of the males and allow a few males to survive. Or those populations will go and mate with other populations. So there are measures in nature that can help the Wolbachia, help the insect combat this Wolbachia.
This takes us back to Jurassic Park, doesn't it? Remember how they had tried to make sure that they would only have a particular gender among the dinosaurs? And Ian Malcolm, played by the inimitable Jeff Goldblum, said, "Life finds a way." And I say the hand of Darwin is on all that lives, but it's absolutely true, right? There's going to be some leakage.
There's going to be some leakage, and there's also, maybe the Wolbachia doesn't express at 100%. So we do get into some gene expression as well, that maybe the Wolbachia can control the expression of this trait so that it may not be 100%.
No, I think this is so fascinating. And again, not every Wolbachia acts as extreme as this. This is the important thing to keep in mind. It's not one Wolbachia. I mean,
Yes.
it is at the 16S level, but you know what I mean.
Right, exactly. It's not one Wolbachia. Not only do they have different genes, different genetic makeup, but they have different regulation of those genes. And so Wolbachia can behave differently. And what's really fascinating is you take one Wolbachia and put it inside a different host, and it can have a different effect on that host. So there's not only different Wolbachia, but there's different host microbe associations.
An emergent property.
Yes, and there's always an evolutionary arms race too, because as the Wolbachia is doing this, the insect then has to evolve ways to combat that Wolbachia. And so you have these two genomes, evolving to survive with one another.
I just think there's so many questions involved in something that you have trouble seeing without a microscope.
Yeah.
It's so interesting to think about the manipulations. And I used to teach a course on this kind of subject to first semester students, and they were deeply disturbed by what they initially thought was science fiction-y, but turns out to be much more the rule than the exception, that some types of organisms can actually affect many aspects of their larger partner. And this is the amazing part about Wolbachia. I also wanted to underscore what you said. No two Wolbachia really seem to have the same effect when you look at the different clades. There are some, like the green miners, that actually make plant hormones, and it's fascinating, fascinating stuff, but there is no one. And that makes it tough to study because there's so much out there.
It is. And I will say, so actually the focus of my research is on the phage. So this is the virus that infects the Wolbachia. And these viruses integrate, yes, exactly. They integrate themselves into the genomes of Wolbachia, and they carry genes within their genomes. And it's actually those genes that are responsible for a few of these manipulations that we've discovered so far. So not only do you have a bacteria that's affecting its eukaryotic or its animal host, but you have the virus within the bacteria that is affecting the animal host. So you have this incredible tripartite symbiosis occurring, which is fascinating from a genomics and an evolution perspective.
I kind of my brain is in a constant state of explosion. Now you know why I call these kinds of things WTM, for what, the microbe, or OMG, overwhelming microbial greatness. Because when you think about bacteriophages out in the environment looking for new cells to attach to, inject their genetic material, and take over, we're used to that. But this is taking place in the cytoplasm of cells. So yes, the prophages are part of that, but it isn't simply a prophage. There are also bacteriophages present. So it really is colonizing the inside of a cell like a niche in evolution, in ecology, I should say.
Yes, it is. It is. And it's really fascinating when you think about it, because this phage, in the environment, phages have to contend with the environmental pressures. But when it's inside a host, not only does it have to find its bacteria, but it also has to contend with the animal's immune system and everything that's going on within that animal host. So it has to be able to get from one Wolbachia to the other to be able to infect the next bacteria. So it's an incredible system, and it's really hard to study because we can't culture Wolbachia. So everything we do we do it within a host. We can't culture Wolbachia to study the bacteria. So it's a very challenging system, which I think people that study Wolbachia and their phages, you get a chance to kind of think outside the box quite a bit, because you can't apply your basic techniques. So you really have to come up with new and interesting ways to approach the situation.
Well, you have to be smarter than them, and they've had a long time to evolve, haven't they? So to me, this is so important because we think about the structure of a symbiosis, and we're used to very peripheral symbioses where you can cultivate both members. And then at the far end, let's look at mitochondria just as an example, or chloroplasts that have fully integrated into the hologenome. You knew I was going to work that in there.
Yes.
And that's your life every day, I'm quite sure.
Yes.
Yes. So anyway, what I was trying to say is this is fascinating to me because the idea of microbes living within organisms that are between those two extremes are really interesting. And I think Wolbachia is a good system for that. Though again, you can't cultivate it
You
yet.
can't. Yeah, and you mentioned something also that's interesting because we talk about the reproductive impact that it has on its host, but some Wolbachia have evolved into mutualists. And so
Mm.
in this case, maybe they encode a novel nutritional like a biotin pathway. And so now the insect has become to rely upon the Wolbachia. So the Wolbachia needs its insect host or its animal host, and it needs its Wolbachia. So we do see this progression. Again, there's many different kinds of Wolbachia. There are mutualists. And it's fascinating. So we like to study the genomes of the Wolbachia to help us understand how and why it's able to evolve these different types of symbioses.
I also wanted to ask, you mentioned that you are talking about Wolbachia within um, I'm going to say the egg producing machinery, the ovaries of the insects. Now, is that always the case thus far? I understand that that's how they get to the next generation, but are other cell types of the host infected?
There are. So we tend to focus on the reproductive tissue. So Dr. Carr in our lab focuses on the sperm. She does a lot of sperm dissections and is looking at the Wolbachia-induced modifications there.
Mm.
But you do, if you look at within an insect, you can find it elsewhere. So you can find it in the somatic tissue. It seems to be more enriched in the reproductive tissue, but you can find it elsewhere. And there are some really amazing studies that are looking at the impact of Wolbachia on behavior, for example, because what if you have, a lot of Wolbachia in the brain or the gut? Like, what is the impact of Wolbachia? And I don't think we know as much there yet.
But that's the exciting part. And there are plenty of people that like to know the answers to everything. And that's great.
Mhm.
And more power to them. They're far more successful than I'll ever be. But I get so excited by things I don't yet know.
Yes.
And that's just me.
Yes.
So what you've talked about, and I just wanted to say for the listener and viewer, and this is obviously nothing, this is all of this more than I do, but it's important to note that it's just not arthropods. In some cases, certain filarial worms and nematodes can be infected, and the Wolbachia appears to control their reproduction. The reason this is important is that there might be ways that we can deal with certain health problems in that fashion. But anyway, I just wanted to say parenthetically that the more we look, the more we see that sneaky symbiont getting into different organisms, right?
Yes. Yes.
I always have students who ask if we have it. And all I can say is that and I actually got so concerned with that, I actually did a search in the human genome. And no,
And
fortunately. No, fortunately. Maybe you folks do since you work with it. That might be interesting, right?
yeah, no, this is sad. I was once doing an outreach event and I was doing it with elementary children. And I've
Mm.
learned now not to talk about the reproductive impact because I had a boy start crying. It's really
Oh.
scary. This little boy when I was talking about the male killing. And it is important to think, I mean, it doesn't infect humans. So
Right.
far, it's arthropods and nematodes, unless you are a human that is infected with one of these filarial nematodes.
Yeah.
But all of these things will not affect us. It is not a symbiont of humans.
So this is actually in heartworms that we see in dogs and cats too.
It is. And if you have had a pet with heartworm lately, you'll probably notice that when you go to the vet, the first thing they do now is give you doxycycline because it is a more gentler way. If you kill the Wolbachia, then it keeps the worm from reproducing. And it's a gentler way of treating these animals because the anti-filarial drugs can be very harsh on them. So the first thing they want to do is get rid of the Wolbachia.
And I haven't even talked about dengue with you. Anyway,
Yes.
there's so much to say about this, but I do want to hear about discover the microbes within. But as I say, I get really excited by this, and I'm carefully watching the time since here's a surprise for you. Sometimes I become verbose.
No.
Ah, ha! But what I was going to say is I'm going to put a lot of links up to all of this in the show notes, and I may ask you for some more updated ones than I might have. But there's a lot of evidence that some blood feeding insects, when they receive new types of Wolbachia, they're less able to transmit certain types of viruses, like the dengue virus. And so the reason that I bring this up, just parenthetically, and we can talk about it more later if you want, is that a lot of people have interest in Wolbachia for the basic biology and others for disease reduction.
Mm-hmm. Yeah, and I think that is important, especially when we talk about the education aspect, because this is a new and emerging frontier. The Wolbachia mediated releases, they are ongoing. They have been very thoroughly tested, and they've been incredibly successful by releasing Wolbachia infected mosquitoes. There's two different applications. One is by utilizing the cytoplasmic incompatibility that we talked about, and you can replace the native population of mosquitoes with one. It's the same type of mosquito, but now it has Wolbachia and it can't transmit disease. And so you're leaving the ecosystem intact. You're not affecting other members of the food web. You're not using pesticides, but you just now have mosquitoes that can't make you sick anymore. And the other is something that is increasingly being used when we see mosquitoes moving into regions, particularly because of climate change, where they don't belong. And they're just messing with the whole ecosystem. They're invasive. We don't want them there. So in this case, we only release the males. And by only releasing the infected males, they mate with those uninfected females and their offspring die. And so that's a way that you can eliminate these invasive species, again, without using any chemicals or other harsh products. So this is a new and emerging frontier, and because we still don't know which insects have which Wolbachia, and there are millions of insects, right? We need help. There's not enough scientists to go out and study the mosquitoes within your backyard or all of the agricultural pests and everything else, so we need help. And so that's how the Wolbachia Project was born. Because not only is it a way to introduce these skills and techniques to high schools and colleges, but it invites them into the discovery process with us. And it really, I mean, now if you think about it, across the world, we have this entire army of scientists that are going out and studying this symbiotic relationship that can then inform us as scientists. So that is how it was started. And we have actually in Hawaii is a fabulous story of one of our colleagues and partners there, because the mosquitoes, invasive mosquitoes, have introduced avian malaria. And there are some forest birds, particularly the honey creepers, that are risking imminent extinction. These birds only exist in Hawaii, and once they're gone forever. And it's because of the mosquitoes carrying the avian malaria. So how do we get rid of these mosquitoes? Well, students went and studied the Wolbachia, they studied the relationship, and they've actually given talks to their local communities, and they have aided in the process of adopting a Wolbachia-mediated release in Hawaii, thanks to students going out and studying this and informing the local community and stakeholders. So it's one example of how students are making a difference in their own communities by doing real science in the classroom.
I can't emphasize how important that is to the listeners and viewers. So many times when students take a course in biology and again, if you hear, I'm not drinking the haterade or eating hater tots with this, but so many times they're demonstrations. And of course the students are bored with it. And sometimes the professors and instructors are as well. So what these are generally called cures, for course-based undergraduate research experiences, which are now moving into the high schools, as you mentioned as well, give students an opportunity to do genuine science, learn genuine skills, and what they find matters, like tiny earth, like sea phages, like evolving stem. These are all wonderful programs that are based on ownership that the students have with things that they find. What's especially attractive about yours is you have a database of everything. And I love your example from Hawaii, and I hope I can find a PR piece on it that I can post for the
Yes.
listeners and viewers.
Yes. Yes, definitely. And I think that's one example. When I moved to Pennsylvania 2 years ago, so we moved our lab to Penn State. And of course, as soon as I moved here, I heard about the spotted lanternfly. I was not as familiar. It's a beautiful organism, but it's wreaking havoc on the forest industry and the agriculture. And so, of course, my first question was, well, does the spotted lanternfly have Wolbachia? What Wolbachia does it have? I did a search and I couldn't find anything online except for Wolbachia Project students because they have been concerned about the spotted lanternfly coming into their community. They have said, could we use Wolbachia? First, we need to know about it. And so they have uploaded data into the database. And that was like the best and only data that I could really find of Wolbachia sequences in the spotted lanternfly. So again, this is really at the forefront. We have students thinking about real world solutions to real world problems and what can they do to contribute to it. So, I mean, I love it. And you can find that in a number of cases. Students are really interested in organisms that are unique to their communities that,
Hmm.
again, as scientists, there aren't enough of us to keep up with it. So we need this.
Well, I tell, and I'm sure you do too. I tell every one of my students that even little children are scientists. They're always making observations, they're testing, and then they're asking others to confirm their experimentation, right?
Yes.
And so, in a way, all of our students at any age are natural scientists until they learn somehow that it isn't cool or interesting to be a scientist. And the one thing that seems to work is something that they find, something that they discovered, especially if no one else has seen it before.
Yes. Yeah. And they live in this world now where we have, you're hearing about climate change, all of this negative, we have viruses. And so give them the tools to come up with solutions. That this makes them, not only are they contributing, but it's helping them their mental state to be able to come up with solutions. They can make a difference in the world. And so they're learning at a young age, I can make a difference and
Yes.
I can contribute to solutions. So I think that's what, like you were talking about Joe Handelsman's project, a lot of these projects are just giving them the tools to
Mhm.
be able to come up with solutions that are affecting their lives.
There's nothing better when they find something new.
Mhm.
I've made the joke many times about what the nature of discovery really is. And it's when someone looks at something and says, that's weird, but that really is how things happen.
Yes.
And I can't emphasize how important that is over and over again. I mean, when a student finds in my own lab, my teaching lab right now, where they'll find a particular isolate that they have from moss that seems to have antibacterial action, and they find out what the genus name of is, and they want to know more. And that's the key. And
It is. Mhm.
they don't have to be a PhD scientist. They don't have to be a scientist at all. They can be a regular citizen, a citizen scientist.
Yes, and I think something if like iNaturalist, that's a very popular tool where again citizen scientists are contributing to our knowledge of global biodiversity. So again, having these online tools is something that we spent some time developing because all of these discoveries are novel and informational discoveries. So we want to create an environment where students can then put all of their data and again contribute to our global understanding.
So I don't know if you want to just, work through what you do and discover the microbes within, or whether you would like to imagine that I have captured an insect and the steps that I would go through.
Great. Yeah.
Which do you think we should do?
Yeah, I think that would be great. So going through the second, you've captured an insect and you're curious. Maybe it's an insect that you've seen buzzing around lately, this new insect. Maybe it's bothering you, eating all your crops. And you're really interested in what types of microbes might be in that insect. Does it have Wolbachia? Well, the first thing you're going to do is bring it into the lab and you're going to do a little bit of digging to try to identify it morphologically. So based on its physical characteristics, try to identify it. What might it be? And then you're going to extract its DNA. So you're going to homogenize the insect, get all of its DNA, And by that, within one insect, not only do you have the insect DNA, but you also have all of the bacterial DNA, all the viral DNA, everything, every organism within that insect, you're getting that DNA. So you have to be able to amplify just the DNA you're looking for. So you're going to do polymerase chain reaction, or PCR. And we are going to look for two different genes. One is a cytochrome oxidase gene that's in the mitochondria of the insect, and it's the universal barcoding gene for insects. So that's going to help us confirm our identification. And then the second gene we're going to amplify is the Wolbachia 16S, which again is another universal identification gene that will help us. Then we're going to run it out on a gel using gel electrophoresis so you can visualize those bands. Yes. And at that point, you'll be able to see, you should see a band for your insect. And then if your Wolbachia is there, you'll see a band for the Wolbachia. If there is no band, it will be absent, and your insect is most likely uninfected with Wolbachia. So that's a stopping point there. That's some really interesting data there. Does my insect have Wolbachia? Does it not? Is it infected or not? If you wanted to go further, you could then sequence that DNA. And we like to sequence both of them. So we sequence the insect DNA because, again, it's a barcoding gene. So that will allow us, when we take that sequence and query it against a national database of known insects, we're able to identify it. And then we will also sequence the Wolbachia gene to be able to tell which Wolbachia it is. Because again, if you're wanting to use something like cytoplasmic incompatibility, you have to have an idea of which type of Wolbachia it is. And so students will do some bioinformatics. They'll build some phylogenetic trees looking at the evolutionary relatedness of those insects with other insects, or the Wolbachia with other Wolbachia. So throughout this process, they're learning a lot of biotechnology and molecular biology skills. But at the end of the day, they're identifying their insect and determining whether or not it has Wolbachia. And I should say, I had one student, I was teaching one course. He uploaded his insect to iNaturalist of what he thought it was. He then had an expert come in and say, "Oh, you're wrong. It's actually this." He was devastated. He wasn't used to taking this feedback. When we got the DNA sequencing back, he was actually correct. That was the proudest day, I think, of his. He just came in with a beam on his face and said, I was right. And he had the DNA sequence data to confirm it. And so this just shows, science is changing, and we have to use special tools in order to make some of these. We make predictions, and then we can confirm it with data. So he confirmed his prediction, and it was just an amazing day for him.
So how did the professional entomologist respond to this, is my question.
That's a good question. I don't know. He went back into iNaturalist, and the student did, and said, no, I think it's this. So I'm not sure how the record played out, but as a learning moment and as a self-confidence moment, it was such a success for that student.
Think about the power of all of this. Mm-hmm. And I love the giant machines that go ping, but I don't have big grants, of course, to purchase the giant machines that go ping. That's a Monty Python reference, which you're probably too young for. But in any event, what I'm trying to say is that this type of equipment is stuff that I certainly have, and a lot of people have, and many high schools have now. And so this idea of using this approach, taking advantage of the online bioinformatics, taking advantage of the catalogs and databases, and especially this business that the student found this, the ownership of it is critical. Now, what I like best, and you're going to think this is no big deal, but I've seen it really pan out, is that when the student is trying to run the gels that you just mentioned, and they don't get a band for the Wolbachia primers, okay, but they also don't get a band for the cytochrome oxidase. They'll look at that, and then they'll say, well, that means I didn't get good DNA. And I'm so proud, that they actually got that part of it.
Yeah.
Because it sometimes they focus, I tell people, the students, you have to squeeze the data like a chamois to get everything out of it that you can. And that negative results are useful too. And of course we want positive results, but the negative ones are important as well. And so this is where the scientific rubber meets the road. You
Yes.
know what I mean?
Yeah. It
And
is. It is. Yeah.
yeah. And this is just wonderful beyond words. And I want to say parenthetically, there are going to be lots of links for the Discover the Microbes Within program that Sarah administers. And well, can I call you the kahuna? I don't know. What would you like to be called about that? You're the director,
The
excuse me.
director.
Yeah.
It actually was created. It was created about 20 years ago with a whole group of scientists and educators and outreach specialists. So I'm just the lucky one that gets to carry it on. But it was a huge team effort, bringing together
Oh,
science and education.
I remember this when it first happened because I'm old. But what I it's true. What I was going to say about it is this website is going to listeners and viewers are going to so enjoy looking at this website because everything's there, and it's really exciting. It's well put together, and it's the kind of thing that I hope you don't mind my saying so. I wish that my sons had experienced in high school.
Oh, well, thank you. Thank
Yeah.
you. Yeah.
So I like the fact you talked about some of the aspects of it and the outreach that you do. So do you have any thoughts about like you've already told a couple of success stories. Do you have anything that comes to mind of another really fun success story about this?
A really fun. Well, I should actually. We do have a student. I mean, a teacher in New Jersey who went to a workshop and met some teachers from Israel. And
Oh,
those teachers decided that they wanted to set up an international collaboration where their students would both look at mosquitoes in the US and in Israel, and then they would get together over Zoom and compare their results. So they set up a whole symposium where they presented to one another. Yes, and we also have some teachers. We have a huge consortium of teachers in Switzerland, actually, that are also looking at this. And they're just like, not only is the Wolbachia novel, but a lot of the Palearctic species of insects haven't been studied yet. They're not in the databases. The databases are very, U.S. centric,
hmm.
and so a lot of the barcodes, insect barcodes, are novel. So we are having some nice collaborations that are being set up between teachers in different schools with different backgrounds. That just shows how global and universal science is, and how collaborative science can be. So,
Can you imagine a student hopping on Zoom to talk to another student in Switzerland about the work that they're doing?
yeah, isn't that crazy?
I think that's wonderful.
Mm-hmm.
And again, what I'm trying to say, I often wear lapel pins that say science is for everyone. And I perfectly understand that there are issues of access, and there always have been. What I admire about what you and some of the other cures that are doing outreach have done is an end run around that by bringing the materials to them. So this is something that can enrich the education of anyone. And I know that you do, forgive me, teacher outreach for like secondary schools. I wouldn't be surprised if you do them for retirees next.
I've had some interest in that, actually. I think that would be fabulous. I, you know, and if I can, I give you one more success story, just one more.
Please.
Okay, so speaking along that term, along that line, I had a father reach out who was not really happy with his, he wanted his daughter's school to have a little bit more molecular biology, so he found us and set up a basement lab and ran the Wolbachia Project in the basement with his daughter because he wanted her to have real hands-on science experience. And which was great, and they had some fabulous discoveries. She then went on to college. She liked the experience but realized it wasn't for her. And that's part of it. I mean, not every student wants to go on to do science, and part of it is learning what you do and don't want to be. And so she enjoyed it but said, I'm going to do something else. However, he said, I always loved science. I had a great time. I think I'm going to go back and get a graduate degree in science. And so he enrolled in a master's program because he made such a fabulous impact on him. And so it is for everyone. Everyone can contribute. And I just love that story about how not only, it helps people decide what they want to do in life, and it doesn't matter where you are, what stage you are, you can always do science.
To carry out the various procedures and experiments that you've discussed on your website. Is there any source of information about what that would actually cost or how that could be done?
Sure. And so on our website, we do have a list of supplies, but I just want to note that there are a few companies that are taking these large, expensive research grade equipment and building them as smaller, more affordable, classroom sized. So one company that we work with very closely is Mini One. There's also other companies such as Mini PCR. So instead of spending $10,000 on a PCR machine, you could get it for $1,000 or less from a place like Mini One. And so if you go to these companies, they're very small. You have smaller sample sizes, but they're affordable. That's how classrooms can get these. Again, it's like Mini One, Mini PCR, and some of the other scientific supply companies. So in the equipment alone, I would say probably $2,000 to $3,000 for the equipment, and then we would supply all the reagents at no cost.
I think that it's important to note as well that I am guessing as time goes on, there are going to be many, many shared spaces that people could use
Yes.
because it all does come down to access.
Yes, it does. It does.
I'm
And loaner equipment. So we have loaner equipment. And again, these community centers will have some have loaner equipment that would allow for more access.
I'm waiting for the Discover the Microbe Within minivan that carries all the equipment around.
Maybe when I retire, that's what I'll do. Maybe we can do it together.
Wouldn't that be fun?
Yeah. Yeah.
I've often heard at my own institution, and I'm sure you've heard the same kind of thing, that again, issues of access and privilege come into things.
Mm-hmm.
And of course it does. And what I like is to find ways to do outreach around that. And you just talked about one. Now, someone could easily say, well, that father had the ability to purchase those materials. I actually know for a fact that a lot of materials get provided. But in addition to that, there are often areas in most communities that things like this can be set up to share with. I don't want to say a community lab, but there are things like that. They're often called incubators.
Yes, or these maker spaces, things like that.
Mm-hmm.
There are some communities that are setting up, or if you have a local science,
Right.
science outreach, yeah.
And I want to emphasize again, and in no way do I want to sound political in any way because I'm incompetent to speak on that subject. But what I would say is when I look around at how science and technology are generally portrayed in the media, there are some happy exceptions, but mostly I see a lot of incomplete and sometimes inaccurate information being spread. And rather than rail about it, the solution is to teach people the scientific method, how we go about things, what constitutes data in a way that really engages them. So what I tell it so happens in the spring semester when I teach this introductory class, most of my students are not biology majors, but every one of them that seems to go through it have gone through the experience of observation, discovery, writing up material, and I think it'll help them. Again, I also point out to them that at the conclusion of my class or at the conclusion of any one of your workshops, the participants know more about biology than most members of Congress. There, I said it. And it's really true. We all can be scientists in our own way. We can be scientists without having the union card to practice science.
Yeah, and I think one of the things that I have noticed about a lot of the work that you do too, I once heard someone say, if you make it to the dinner table, that was a success. And I
Yes.
think if you introduce these concepts and these skills and they go home and talk about it to somebody else, that is a true measure of success. And I know that your classes are particularly good at doing that. So thank you for all that you do and just spreading the word of microbiology, because in doing so, it can then grow exponentially and increase our scientific literacy.
As microbes do.
Yeah.
I admit freely, I wish I could claim the title of microbial evangelist, but there we go. I do my best with this. because of all the negativity that we all hear about. But in any event, where do you think Discover the Microbes Within will be going in the next few years?
That's a great question, and it's something I'm working on now, actually. I'm really excited about it. But now Wolbachia is great, but there's the whole rest of the microbiome that still needs to be studied. So we might expand it. I mean, Wolbachia is always going to be our favorite. It is the coolest microbe out there, but we might expand it and look to see how all of these microbes are playing around. And we can also begin to incorporate more next-gen sequencing because now, as you see now, a lot of schools are starting to have molecular biology equipment. I'm starting to see now they're starting to buy their own sequencers, which blows my mind. And so we're thinking about how can we now bring in larger scale sequencing into the classroom.
I think that this is so important. I mean, one of the silly things that you no doubt know that I do is I have students isolate organisms from their water bottles.
Mhm.
It would be very interesting to do the next gen on that to figure out the non-cultivable members that are in there as well. But that's a little price prohibitive right now. But
It
bringing
is.
those bringing those prices down would give them, here's what I isolated and here's what the whole population looks like. Think how that opens up the mind.
Yes, and I think we're starting to see the prices come down already. So it is within the foreseeable future that this could be part of the school curriculum.
Oh, I so hope so.
Yeah.
Well, I really appreciate that. Are there and I now would toward we're getting toward the end, and I wanted to say, are there other things that you're excited about or want to tell listeners and viewers about?
Uh, I think, yeah, I think I just keep being engaged and keep asking questions. And I am approached all the time by teachers with just these wonderful, amazing ideas that I don't know how to answer. You know, we have teachers looking at ticks, for example, and they're looking
Hmm.
at Lyme disease and Wolbachia and looking at the co-infections. You know, keep coming up with these great ideas and let us know because these areas of community interest, we need to know about them so that we can help to facilitate more people studying this and enable people to do that. So I just want to really have an open door with people. I want to hear what they're interested in and how can we help.
So again, as I have said before to the listeners and viewers, I'm going to be putting lots of links up, and I so strongly urge you to have a look at this program the way that I've told you should look at another program that I'm used to, Tiny Earth, as well as Evolving STEM.
Mhm.
All of these are things that are so exciting to read about and think about. And I know how hard you and your entire crew have worked on this. And it must be so gratifying to see it work the way it has been.
Yeah, thank you. Thank you. And it's taken a while. You have to I think part of it if you're having a project like this, you can't just put it out for a few years and walk away. You have to find a way to just keep it going. And the longer you have it going for us it's been 20 years. The longer you have it going and the more it'll grow.
You know, there is a wonderful program called the C-phages program, which
Yes.
is wonderful. It
Yes.
involves isolating bacteriophages from nature,
Mhm.
characterizing them, but it's all predicated on having an electron microscope, at least for part of it.
Yeah.
And since that not everybody has access to that, there's a limitation there. I'm not saying it's impossible, and I tip my hat literally to the people that work in that program.
Mhm.
I don't know if you know this, Sarah, but they had to institute, apparently, this is what I've heard. The students get to name the bacteriophages that they
Yes.
isolate, and they had to institute a rule that they could no longer be named after Nicolas Cage.
No. Oh, that's awesome. I love
Too many
that.
cage phages, right? Yeah.
That's
And the other thing that I think will start to happen in the future are how organisms interact with one another. And we're used to that idea in ecology, and it's certainly true in the microbial world. And we've been so reductionist in the past. And I've seen the more ecologically oriented students in my own classes just light up when we start talking about those kinds of things.
yeah.
So this is important work that you're doing. And again, I want to salute you for it.
Yeah, thank you. Thank you so much. And I should say I see them light up when we talk about computer science. You know,
Yes.
we have some students that are like, "Oh, this is fun. This is cool." You get to the bioinformatics and a light bulb comes on. And so that's really exciting just to see, different students' interests can peak at different parts of the process.
Right. And again, it comes back to ownership, right? That
Yes.
they did something. They're not like just following the protocol. They're actually looking at what they get. Even when they do bioinformatics, they're looking at it and thinking about it.
Yes.
Are you going to be at Microbe in June?
Maybe. I haven't decided. Are you?
Well, I am going to be there, and my wife Jenny's going to be there. So if you're there, dinner's on me.
Oh, well, thank you. Thank you.
I've really enjoyed our conversation. Would you please give my very best, to your family and to your spouse?
Yes, to my other half, Seth, I absolutely will. Yes,
Yeah.
thank you.
And I want to wish you a wonderful, wonderful day.
Thanks, you too. Thank you so much for having me on. This chat has been so much fun.
Thank you so much, Sarah.
Yeah.
This has been Matters Microbial, a weekly podcast about the wonders of our microbial world and the people who study it. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. Show notes from today's episode with indispensable links can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department of the University of Puget Sound in Tacoma, Washington. Professor Sarah Bordenstein is in the departments of biology and entomology at Pennsylvania State University. Many thanks, as always, to David Renata for superb editing and Reber Clark for the wonderfully quirky music that brings a smile to my face each week. I hope you've all enjoyed being part of our Quality Quorum for today. See you next time on Matters Microbial.